Hi, AP Bio folks. I want to talk just real briefly before we start today about the assessments that you've been taking in this class. As we move into second trimester, you'll notice first of all that on the College Board assessments, I'm adding a timer. That's to help you get used to the amount of time that you would have on the actual AP exam. It doesn't cut you off at the end, but keep in mind if you're going way over that time, you need to work on some strategies to help speed up your ability to process those multiple choice questions so that you don't run out of time on the exam. And we're going to start talking about some of the multiple choice questions during some of the live sessions. And so if you see that on the calendar, those are good days to try and show up to get some of those tips and tricks. For the assessment you took right before break, your scores will be released once everyone has finished taking that assessment. So that's not quite yet. There's still one or two people taking those multiple choice questions, but I'll release those scores when it's done. There was one question this time. I looked through all the scores and there was one that only about 25% of people got it correct. And so I decided to throw that one out. When I see that, that usually tells me that question was either too challenging or too tricky. And so all that means is you keep the same score. If you got 6 out of 8, you still have 6 out of 8, except now it's really 6 out of 7. Okay, so in the gradebook, that means this will have a total of 9 instead of 10 because there were 2 for that free response. In these free response type questions like this assessment that we did, many people on this one used the diagram from the multiple choice. They used the information in that right answer to help you figure out why these were wrong. But in the future, I might not even show you the right answer because the point of this really is to be able to look at these others and figure out why that can't be the right answer, to be able to eliminate some. So when you read letter B, what you'll need to do is just read that and figure out, okay, what part of this is wrong and why can I rule that out? And you don't need to talk about or understand everything about it. You just need to be able to find a word or a couple words that tell you that this one can't be the right answer and explain that. So we're going to continue practicing these because this is a really important skill in your ability to take multiple choice questions in AP Bio. If you have any questions about those assessments as you get them back, please make sure to let me know. All right, now we're going to go back to chemical reactions. So we talked mostly on Tuesday about reactions in terms of energy. Now we're going to put that together with the processes that are actually happening in cells. We divide chemical reactions, the process of metabolism, which is both building and breaking, into 2 parts. We talk about anabolic reactions. Anabolic reactions are things that require energy to build larger molecules. So you can see examples here of like combining amino acids to make a protein or combining a glycerol and fatty acid to make a lipid molecule. Those are anabolic reactions. Anabolic reactions build molecules. And the way that I remember this is when I see this capital letter A, I think of it like a hill. We're taking little things, we're putting them together to build something bigger. And that type of reaction requires energy. And some examples of this are things like dehydration reactions. If you remember those from a long time ago in our chemistry unit, dehydration reactions are when we kind of pull a hydrogen off one and an oxygen and hydrogen off the other to make a water molecule. And in the process, that forms a bond between those 2 monomers. Photosynthesis is another example. We take small molecules like carbon dioxide and water and attach them together to build something bigger like glucose. These all require an input of energy, and so they don't happen spontaneously. And that energy that we put in is stored in the chemical bonds that are formed between those atoms. These are examples of endergonic energy is going into the molecule. And we saw this diagram yesterday. So endergonic reactions, the amount of energy stored in chemical bonds is increasing, which means delta G, that change in the free or available energy, is greater than 0, is a positive number for these types of reactions. The opposite of an anabolic reaction is called a catabolic reaction. And you can see here we're starting with a big molecule and we're breaking it into smaller and smaller pieces and releasing energy along the way. The way that I remember this one works really well for anyone who has a pet cat. My brother had a cat once that was like famous for waking him up every single day by walking across his desk or his shelf and knocking off everything that was sitting there. Cats break things. And so if you have a cat that likes to do that, and you can remember that cats break things, then you can remember that catabolic reactions are breaking things down. So catabolic reactions, we start with a large molecule and break it into smaller pieces, which releases energy. So again, some examples of this are hydrolysis. Remember, hydrolysis is the opposite of dehydration. That's where we put the water back in, which breaks that bond and separates it into its component parts. Or something like cellular respiration, where we take a big molecule like glucose and break it down into smaller pieces in order to release energy. So this is an example of an exergonic reaction. The energy is exiting that reaction. And this is the graph we looked at yesterday. You can see in this case, there is less free energy stored in the bonds of the products. So the free energy within the molecules, the available energy is decreasing. So delta G in this case is less than 0. Delta G is negative. So living things require both catabolic and anabolic reactions, and they're very closely linked to each other, and they work together, and they're connected through this molecule. Take a minute and look at this. You might even want to pull out those pink packets from the biochem unit way long ago, where we looked at diagrams of molecules, and see what parts of this molecule you recognize. Hopefully there are some pieces in here that looked familiar to you, and you could at least kind of take a guess at what this is. This is actually a molecule of ATP, which is something that I know you've heard of and is really important in linking together catabolic and anabolic reactions in living things. So this picture kind of shows how these all go together. So we take in molecules through our food and break them down as we digest food. Digestion is a catabolic process, right? We're taking these big molecules in our food, we're breaking them down into these little pieces. We're breaking our food down into simple sugars, into nucleotides, into amino acids, and other small pieces that we can then use in anabolic building reactions to build the molecules that we need for our cells. So you take in protein through the meat that you eat and dairy products you eat, things like that. You break apart those proteins. Your body recycles those amino acids to build the enzymes and the proteins that your cells need to function. In those reactions, during that breaking down of your food, of course, some of that energy is always lost as heat. None of these processes are totally efficient. But the other thing that happens as those are broken down, breaking things down releases energy. Some of that is lost as heat, but some of it is captured in a useful form. And that useful form is generally the molecule ATP. There are other useful forms of energy. Occasionally, you'll see one called GTP, which is another kind of energy storage molecule. But here we're focusing on ATP because that's the primary intermediate molecule. Really, the job of ATP is to capture that energy that's released in this breakdown of food and other molecules so that it can be used to keep it in a usable form so we can use that energy to build molecules on this side and carry out those reactions that we need. That's the job of ATP in our cells. So at the bottom left of the slide, you can see that diagram of ATP that you just looked at. And hopefully you identified a couple of pieces. There is a ribose sugar. That's this component right here. When you looked at this, even if you couldn't call it ribose, hopefully you recognize that as a carbohydrate. It's this carbon ring. Remember, there's carbon at each corner, connected to some oxygen and hydrogen. Carbohydrate, carbon, oxygen, hydrogen. That's all we've got there. So that's carbohydrate. You also, up in the upper right here, have the base adenine. This is actually one of the nitrogenous bases that you find in nucleic acids like DNA and RNA. That's that letter A of your A's, T's, C's, and G's. It's got a bunch of nitrogen in those rings. That's what shows you that it's a nitrogenous base. And then it's attached to one of those functional groups that we learned about last trimester. This is a phosphate group. Now, hopefully, this kind of rings a bell. If we have a sugar, a phosphate, and a base, this right here is a nucleotide, like a building block of a DNA molecule. ATP is actually a modified nucleotide. It's the same type of molecule that makes up DNA and RNA, except here we've got 3 phosphate groups. And the bonds between these 3 phosphate groups help to store the energy in this molecule. That's the part that we can add and break off pretty easily. And one of the reasons that this bond is good for storing energy is that these phosphate groups are all negatively charged. And if you remember, negatively charged things will repel other negatively charged things. And so it doesn't take too much energy to break this bond because these 2 are already trying to kind of push away from each other. And so it breaks off fairly easily, and the energy in that bond can be released. And so this molecule of ATP is really good for storing and transferring energy, which helps us connect exergonic processes, so capture the energy that comes out of those exergonic processes and use that energy to power endergonic processes that require energy. That connection of exergonic and endergonic reactions is called energy coupling. And we can see that kind of summarized in this diagram here. So if we start with an exergonic reaction, so this would be something like digesting your food. So as we break things down, that releases energy. That energy then, so this is exergonic, it releases energy. That energy then is used to take an ADP, this only has 2 phosphates on it, and connect it to a single phosphate. That's just an inorganic phosphate floating around in the cell. So we're taking smaller things and putting them together to build a molecule of ATP. So if we're building something here, this reaction going from ADP and phosphate to ATP is anabolic. I'm just going to put real brief abbreviations here. And it's also endergonic. It requires an input of energy that we just got from breaking down our food. So that anabolic endergonic reaction builds ATP, and then that ATP can travel wherever your cell needs it and power lots of different cell processes, because then when we break this last phosphate back off, we're taking a large molecule and breaking it down. So this, going from ATP back to ADP, is a catabolic process that's exergonic. It releases that energy. And that energy can then go to help power other endergonic processes to build the molecules that your cell needs. So this is actually the exact same diagram that we just saw a slide or 2 ago. Sorry, I forgot a letter. B-U-I-L-D. But now we're focusing a little more on that role of ATP and how this molecule can be recycled over and over to help link the catabolic processes in cells with the anabolic processes on the other side. So there are a couple of slides in your packet just to allow you to practice and kind of put together all of this vocabulary. Take a minute. You have a whole list of questions like this. Look at the chemical reaction at the top, and for each one, circle the correct word. In this case, are we building or are we breaking down molecules? Go down the whole list, do the whole slide, and then we'll talk about this real quick. So in this case, you're starting with a large molecule and breaking it into some smaller pieces. So this one is breaking down molecules, which means we're decreasing the complexity. These are simpler, smaller molecules. And since we're breaking things down, this is a catabolic process that releases energy. And if it's releasing energy, that's called exergonic. And we are, in this case, increasing the entropy, the disorder. Remember, small molecules are less organized. They're more scattered around than if we have them all connected together. And so the delta G in this case would be negative. There is less energy stored in these chemical bonds because there are fewer bonds here than there were in the original molecule. Because this is an exergonic reaction, it happens spontaneously without any input of energy. The other one that you have here is actually the reaction for photosynthesis. So we take carbon dioxide and water. There should be pluses and arrows in here with an input of sunlight to make glucose and oxygen. So same thing, go through and practice this one. This one you hopefully realized was the exact opposite. Here we're taking small things, carbon dioxide and water, and using them to build larger molecules of glucose. So this should have all of the opposite answers. It needs to be coupled with ATP. ATP helps to take the energy from these reactions over to here to build those molecules. So last bit today to kind of wrap this up. The reason this all matters is that in a closed system, so a closed system means like an enclosed beaker or test tube, something where nothing's coming in and nothing's going out. Chemical reactions eventually reach equilibrium, and the change in stored energy and free energy equals zero. However, in a cell, if there's no energy being released and no energy being used, that means none of those chemical reactions are happening. The cell's metabolism has stopped. And that actually means that that cell is dead because, living things need to be taking in materials and energy and using them to build things and maintain homeostasis as they carried out all those processes. So equilibrium is actually not a good place for living things to be. And they're never there because living things are not closed systems. They're open systems that are constantly taking in oxygen, nutrients through our food, releasing waste products, all of that kind of stuff. So living things never reach equilibrium, but that's a good thing. That's what makes us living creatures. Yesterday, you started applying some of these ideas to the survival strategies that different organisms have evolved. Living things can only take in and metabolize so many materials, and so we have a set amount of energy to use. And living things need to use that energy for a lot of things. Like you read about, we need that energy to maintain homeostasis, to keep our bodies warm, to grow, to reproduce, to care for young, all of the things that living things need to do. But since we only have a limited amount, living things need to kind of make trade-offs. And the way that they have evolved their life history strategies, how they maintain homeostasis, shows some of those trade-offs. And these aren't good or bad. Some living things spend a lot of energy on having tons and tons of offspring, but the trade-off is that they spend very little energy on caring for those young. That's a trade-off that allows them to have enough offspring that at least some of them will survive. So there's an advantage to that. There are advantages to all of the different strategies, but it's important to realize that different living things use their energy differently. You have a few more days to continue working on this application assignment, so make sure that you let me know if you have any questions as you continue working on that. This unit contains a lot of vocabulary and a lot of detailed concepts, so please make sure you're spending some extra time reviewing and studying. We just wrapped up most of chapter 6, except for the last little bit that's about enzymes. We'll come back to that later. There's also an optional vocab summary sheet attached on today's classroom post to just help you put all the words we've been talking about in one place and kind of review. And it makes a good reference for later when this comes up again. We just talked about the energy budget assignment due Sunday. You also have a mastering biology assignment. Keep in mind this one is a little bit longer because we're doing some more complicated topics. Please don't leave it for the last minute or you'll have a lot of work to do. So start working on that over the next week or so. Let me know if you have any questions. Happy studying and have a great afternoon.